delineation we'll talk about very briefly, but we'll get into the specific details of the story. I'm actually going to spend a lot of time talking about the biology of salmonids that's relevant to this particular example and some of the background with respect to that, but it fits into what you guys were talking about this morning already. So basically, the Endangered Species Act. Does anyone have any questions before I get started? And yeah, okay, we'll just see. And I can certainly talk about methods and RAD and other sequencing of DNA and this kind of thing after this. But I think this is a great story for this class. So, is it possible?
Oh, better.
Good. Okay. So basically, as you guys were talking about this morning, the Endangered Species Act provides this legal framework to protect both species as well as distinct population segments below the species level, right? But the problem is the Endangered Species Act itself doesn't define what a distinct population segment is. So basically, if something is considered a distinct population segment, it has full species level protection under the Endangered Species Act, right? And a lot of actions are taken to prevent the extinction of that unit. But basically, what is a distinct population segment and how do you define one? The Endangered Species Act really doesn't give guidelines and doesn't explain how to, or what a distinct population segment is, okay? So it turns out that actually genetics is very, very commonly used it's a really common and often exclusive tool that's used for defining distinct population segments, okay? So defining these units for legal protection under the Endangered Species Act. And basically what's done is, markers from random regions or just different regions of the genome are analyzed, and basically population structure is characterized, okay? So what happens is, for example, in salmon, and this is an example here of salmon populations in Northern California, Chinook salmon populations in Northern California. Basically, there's many rivers, and each river has tributaries, right? So there's big rivers that lead to smaller rivers that have little streams, and so what's a unit there? And so basically what's done is individuals are sampled from across the range, and units, for example, a creek might be considered a population, and these individuals from that area are genotyped, and individuals from across the range are genotyped, and population structure is characterized. And basically what you find is that in salmon and many other species, you find that rivers that are relatively close to each other tend to be genetically very similar, but locations that are geographically distant from each other are distinct, and you basically can group hundreds of different rivers and creeks and streams into these units based on the overall genetic structure of the population, of this large group of fish spread across an area. Okay, so basically, population structure is characterized, and it's used to define units for legal protection. Okay, so it turns out that within one of these units, different rivers, fish from Chinook salmon from different rivers within those units are genetically very similar to each other, but when you look at locations between different units, the populations are much more distinct from each other, okay? So this is sort of the framework that's used to define DPSs, basically for legal protection. Now this makes a lot of sense. It's very satisfying because, locations that are close together tend to have similar environmental characteristics, and certainly, this is somewhat sufficient for protecting adaptive differences between populations, right? So here in California, you can't see this, but it's like a rainforest here. Here it's in the valley, it's much drier. It makes sense that there'd be major adaptive differences between populations from these different regions, and by defining different units, we're protecting those adaptive differences that are important for these fish living in very different areas and very different environments, right? Okay, but it turns out, I guess you guys experience this somewhat here. I'm not completely familiar with the weather, but on the West Coast, in California and Oregon, where I'm from, we have this really extreme seasonal variation throughout the year with respect to precipitation, okay? So it's basically what's called a Mediterranean climate, where in the summer, it doesn't rain at all. Basically, it goes for months without raining, and then in the winter, it rains all the time, right? So throughout the year, there's really extreme seasonal variation in addition to this geographic heterogeneity that I talked about in the last slide, okay? So basically an example of that, this is the Umpqua River in southern Oregon, and an example of that is this creek called Steamboat Creek that's a tributary of the North Umpqua River, okay? So this is a picture of Steamboat Creek. So in the winter, Steamboat Creek has a lot of water, high flows. This isn't right after a big storm or a flood or something like that, but there's just, there's a lot of water, it's cool, and, but this exact same location in the summer, after it hasn't been raining for months, is really drastically different. So the environmental conditions in the summer are very, very different from in the winter because there's extreme seasonal variation, right? So there's very little flow, the water is very warm. The water actually almost, stops flowing basically at the end of summer, so September, for example. And so, again, there's very, very different environmental conditions throughout the year. And what's really interesting is that salmonids, in particular Chinook salmon and steelhead, two species of Pacific salmon, have evolved adaptive differences to take advantage of these seasonal environmental conditions, okay? So basically, the North Umpqua and this river that I was just mentioning, it contains what are called distinct run timings for both steelhead and Chinook. And these distinct run timings are really great because what I mean by run timing is I mean the timing of adult migration. So basically salmon, many of you are probably familiar with this, but basically salmon are born in freshwater. As juveniles, they migrate to the ocean. They spend, a year or 2, 3, 4 in the ocean, grow big, and then return to the freshwater. And basically what this graph shows is this is adult return over a small dam on the North Umpqua River. And what you see is there's 2 peaks of adult migration, okay? So these are bi-weekly counts of adults migrating from, this is June, July, August. So what you see is there's a lot of adults that migrate in the summer, and then you have no migration sort of in the fall and early winter, and then you have a lot of migration happening again in the winter, okay? Now these are what are called summer steelhead. These are referred to as summer steelhead, and these are referred to as winter steelhead, okay? And what's really interesting about these summer steelhead is, so I'm going to use some different terminology here. Basically, this data is for steelhead, but it turns out Chinook salmon, a completely different species, also has variation in run timing, right? Where you have these early migrating individuals and later migrating individuals. And there's a lot of benefits to this. One of the benefits is that it provides fishing opportunities throughout the year, okay? There's ecological benefits. I don't want to go into all the details. We can certainly talk about that if you're interested. But basically, I may be referring to these as early run individuals and these as late run individuals, okay? But what's really interesting about these early run individuals is they migrate up in the summer, but it turns out when they migrate up, they don't spawn immediately. What they do is they get up to the headwaters of these rivers, and basically they hunker down for months, surviving in this basically extreme conditions of almost no flow, okay? And during that time, they don't eat anything. There's no water in the river. They're just basically hunker down in a deep pool and sit there for months before spawning at a similar time to the late migrating fish, which are called winter steelhead. So basically, these fish enter freshwater with immature gametes, get up into the headwaters of a river, hunker down in these deep pools, spend months there developing their gonads, not eating, but developing their gonads before spawning. So although they're migrating now, they don't spawn till much later. So basically, this is a very different life history than more typical late-run individuals that enter freshwater with mature gametes and spawn almost immediately after entering freshwater. Okay? Does that make sense? So basically, let me show you what that looks like. Oops. It basically looks like this. So this is actually a picture of a particular pool on Steamboat Creek up the North Umpqua River. These are large adult steelhead, they're 10-pound fish. And basically they sit here doing this for months, okay? And so the reason they do this is because the terrain, the river, is actually quite steep. And it turns out there's these rapids that are very difficult to ascend in most flows. So by migrating up earlier, these fish can actually get to upper reaches of the river that are difficult to access later in the year when the flows are higher. But to do this, they have to actually migrate up on the snowmelt recession, but then they get there, they have to survive for an extended period of time. Okay, so these fish are basically have this really unique physiological and behavioral adaptation to be able to survive for months in low water and a relatively warm environment. They basically have to store a whole bunch of fat before they enter freshwater to be able to survive, not only survive, but mature their gonads during this extended time in the freshwater. Okay? So from a fishing perspective, actually these early run fish are considered by far much more prized than more typical mature migrating fish, these late run fish, because of the fat content. They taste so much better. For example, they have a much, much higher fat content that allows them to survive for an extended period of time in the freshwater. Okay? And so the behavior and physiology of early run individuals is dramatically distinct from late run individuals. Now, again, let me say this one more time. There's two species that actually have this early run life history polymorphism, Chinook salmon and steelhead. Okay? Now, so it turns out that because these early run populations have an extended reliance on freshwater, they hunker down in these deep pools for months. They've been pretty much extirpated from the entire range. Okay, so things like dam building blocks the upper reaches of these freshwater rivers, and these fish can't access that habitat that they need to over summer to survive. Okay, so a good example of the really dramatic declines of early run individuals is the Klamath. So this is the Klamath River in southern Oregon and northern California. It's a large river. It was once the third largest salmon producer on the west coast of the U.S., behind the Columbia and the Sacramento, which is a little bit south. But basically, actually, historically, spring run Chinook, these early run Chinook, were the dominant life history polymorphism within the Klamath. So it's thought that there was, hundreds of thousands of spring Chinook. A couple hundred years ago, every year, hundreds of thousands of adult spring Chinook would return to the Klamath, and maybe a couple 100,000 fall run Chinook, late run Chinook would return. Okay, so early run Chinook was actually the dominant life history form in the Klamath River a couple hundred years ago. Now in the Klamath, the late run individuals that enter freshwater with mature gametes and spawn immediately, they're still relatively healthy. There's 100,000 or so adult fall Chinook that return each year, but spring Chinook now have gone from hundreds of thousands of spring Chinook to a couple hundred wild spring Chinook in the entire watershed, and they're restricted to one location, which is the Salmon River here. There's sort of one small remnant population of adult spring Chinook in the entire Klamath. Okay, and just as a sort of a note, there's tribal fisheries were huge. There's still tribes that fish this. Historically, the tribes that fish the Klamath focused their harvest on the spring Chinook. They actually didn't, it'd be this long winter. All of a sudden, these incredibly tasty, incredibly fatty fish would start swimming up the river. They would harvest them like crazy. It's a huge part of their culture to harvest these early run individuals. But now basically the early run individuals are non-existent. There's a couple hundred. On a good year, there might be close to 1,000 wild early run individuals in the entire Klamath. They used to be spread out throughout the whole drainage. Now they're restricted to one tributary. Okay, and the same thing is true up and down the west coast. So most rivers that used to have early run populations don't have any at all anymore. But a few places still have small remnant runs of spring Chinook and summer steelhead. Okay. Now, what is being done? Okay, I'm going through this in a little bit different order than. Yeah. Okay.
Why did you say they were restricted? Because of dams?
Yeah, so dams, water withdrawals, logging. So basically they have to survive. So it's hot in California, right? They have to survive a hot summer in almost no flow of water, okay? So things like logging practices that remove shade from the river, water withdrawals for agriculture, dams that block the coolest upper habitats, these types of things have caused this really dramatic decline of these early run individuals. Okay, good question. So basically, actually, many geneticists, so this is fascinating from a genetics perspective. This is a very, these early run fish are very distinct from a behavioral and physiological perspective. And just from a basic genetics perspective is what's causing them to be distinct, and what's the evolutionary history of that? How did these early run populations evolve, and when did they evolve? So what's the genetic and evolutionary basis of early run timing in salmon? I think it's an interesting question that people have been addressing for a long time. And actually the first person to address this question was Fred Allendorf. So Fred, in his dissertation, actually was the first person to genotype early and late run populations and try to look at the genetic structure. Okay? And so basically many studies have investigated the genetic and evolutionary basis of early run timing. And basically, if you were going to approach this, there's sort of two hypotheses with respect to what you might expect to find if you were to genotype early and late run populations. Okay? So one possibility is that when you look at the genetic structure, when you look at early run populations from different geographic locations, they're all genetically similar to each other, and all late run populations from different geographic locations are all genetically similar to each other. So for example, if A is late run populations and B is early run populations, you might do some genetics and find that late run populations are one monophyletic group and early run populations are one monophyletic group. Okay? Alternatively, you could genotype early and late run populations from different geographic locations and find something like this, where pairs of early and late run populations in the same geographic location are genetically similar to each other. Okay? And so the overall genetic structure relates to geography, not to run timing. Okay, so these are two possibilities with respect to the genetic structure of early and late run populations across a broad geographical area. Okay? And it turns out many people have addressed this. The first person was, again, Fred in his dissertation at UW. And basically every one of these studies has found that the genetic structure is primarily influenced by geography as opposed to run timing. Okay? Now that's been interpreted in this way. Basically, if the genetic structure was like this, where you had early run individuals and late run individuals as monophyletic groups, that would suggest that there was a single evolutionary event where late run timing is the ancestral phenotype, where the early run timing evolved once, and then that new early run population spread around different locations. Alternatively, if you find a genetic structure like this, that would suggest that there's been multiple evolutionary events. So early run timing has evolved independently in these different locations. Okay? And it turns out this is the genetic structure that you get. And again, that has been interpreted to suggest that basically there's parallel evolution. You have multiple evolutionary events producing early run timing. Okay? Does that make sense to folks? Okay? So, any questions? Please ask questions if you have them. I'm happy to answer questions. Very comfortable with that. Okay. Now, basically, so all the studies have found that, and I think this quote from Robin Waples. So Robin Waples, who is someone you've talked about 10 years ago, published a paper looking at this. And this is the quote from that paper. Okay? And so Robin says that although the failure of most stock transfers indicates that local populations may be largely irreplaceable on human timeframes, at least some patterns of Chinook salmon life history diversity appear to be evolutionarily replaceable, perhaps over timeframes of a century or so. The evidence for repeated parallel evolution of run timing in Chinook salmon indicates that such a process is likely, provided that habitats capable of supporting the alternative life history trajectories are present and sufficient robust source populations are maintained. So basically, the genetic structure has been interpreted to suggest that this is a very evolutionarily plastic thing. It's very easy for early run populations to evolve from late populations because it's happened many times in many independent locations. Okay? So this has been interpreted, although it's a very dramatic physiological and behavioral adaptation, it's very evolutionarily plastic. And it's happened lots of times. And basically this has important conservation implications. Basically, here's an example. So you have these people who are interested in saving early run populations, but at the same time, the sort of science tells us that early run populations, although they're cool, we want to have them around, if they're gone, it's such an evolutionarily plastic thing that we can get them back, okay? So this is an example, this is a petition that someone wrote, that a group of conservation organizations, NGOs wrote to, their petition was to provide legal protection for spring run Chinook salmon in the Klamath Basin, okay? And basically they say, these groups, we formally request that the National Marine Fisheries Service list Chinook salmon in the Upper Klamath Basin as threatened or endangered under the Endangered Species Act. And they say do this under one of the following alternatives. They say either list spring run Chinook as their own evolutionary significant unit, list spring run Chinook salmon as a distinct population segment, or list the currently recognized ESU that contains both late run Chinook and early run Chinook. List that primarily based on the fact that spring run Chinook have severely declined within this ESU. Okay, but basically, so NOAA looked at this petition. They said, okay, should we provide independent legal protection for early run individuals since they've declined so dramatically, since they're on their way to being completely extirpated? And they basically said this. They said, no, we're not going to do that. And the reason they said no, which is I completely understand it, is because although in their response to this petition, they said, we appreciate early run individuals. We want to have early run populations. However, it's just not really an evolutionary significant difference. If early run populations are gone, based on the science, it's so evolutionarily plastic that they can just come back. So we don't really need to protect them, because if we restore habitats in the future and these kinds of things, they'll just come right back. Okay, is basically the argument. And it's based on genetics. So geneticists have made that argument based on patterns of population structure, patterns of genetic variation that you see across the area where early run populations are. Okay? So go ahead, Gordon.
Yeah, do you know in this case the difference between a DPS and an evolutionary significant unit?
Yeah, so basically NOAA's interpretation is, NOAA uses the evolutionary significant framework in Pacific salmon, but they say that, well, we don't really care if you could list them as a DPS, and from a legal perspective, it's the same thing. But basically NOAA, as far as I know, I'm not a policy expert, but NOAA just uses the evolutionary ESU concept to make listing decisions. Okay, but the people that were requesting that, they don't really care about that. Just one of these three listing scenarios would provide legal protection that they were looking for. Okay.
DPS is typically used for terrestrial, and ESU is typically used for marine.
Yeah,
and they're more or less the same.
Exactly. That's basically, it's because my understanding is U.S. Fish and Wildlife's policy is to sort of use DPS. And there's some subtle differences, but basically they're the same thing. It's more of a sort of, Robin knows all the intricacies of these types of things, but from my perspective, it's just either you're going to call it an ESU or you're going to call it a DPS, and it doesn't matter that much. It's not that simple.
It's a little unfortunate for students and everyone that they have these two terms, but as an example how DPS is typically used for terrestrial, we've talked about grizz and wolves in here, but trout are more inland, and DPS are used often for trout, and since steelhead and rainbow, whereas for the marine, the salmon is ESU.
There's two agencies that sort of implement the Endangered Species Act, is my understanding. One is U.S. Fish and Wildlife Service, and the other is NOAA. And NOAA uses an ESU policy, and U.S. Fish and Wildlife uses a DPS policy, but basically they're the same. The criteria are very, very similar. Okay? So basically, why did they decline that petition? They declined it because of this. Again, this is Robin's quote, and I've highlighted some of the language from this quote. Basically, this Chinook salmon life history diversity appears to be evolutionarily replaceable, perhaps over time frames of a century or so. Okay? Basically, again, the idea that is the science tells us that it's a very evolutionarily plastic thing. So although we want early run individuals, we want to protect this, we just can't justify independent legal protection in this case because it's such an evolutionarily plastic thing. Okay? And so basically these early run populations continue to be, they're on the brink. Okay? But it doesn't matter because they could just come back potentially. Is the argument for not providing protection. Now, basically, this is when I started to get into this. About I don't know, almost 10 years ago now, these amazing new technologies came out, and some technologies that I developed were combined with our massive increase in sequencing capacity, and basically they allow us to approach these problems in a new way. So instead of just looking at overall genetic structure in populations, what we can now do is we can interrogate the genome of virtually any species at an incredibly high resolution. And what that allows us to do is actually find the genes that are controlling these adaptive differences that we can observe. So instead of just looking, basically quantifying the amount of connectivity between populations, now we have the potential to actually identify the genes that are making different populations adaptively distinct. Okay, and so I was sort of trained in molecular genetics in model organisms like fruit flies and stuff like that. Okay, and I was trained as a gene hunter. So in fruit flies, you make mutations and you go out and you map and you find the genes that are controlling developmental processes and things like this. Okay. And so as these technologies made it more and more feasible to interrogate large amounts of DNA in virtually any species, I said, well, I can start working on salmon, and now I can gene hunt in salmon. I can find genes that are controlling interesting phenotypic variation or adaptive variation in natural populations of salmon. So I'm from Oregon. I really like salmon. I grew up fishing for salmon. So I said, I want to start working on salmon. Okay. So basically, when I started working on salmon, this study, so I grew up actually in one of the rare places that has early run populations, and it has an early run population that's supported by a hatchery. So it really doesn't have, the river that I grew up fishing doesn't have wild early run individuals, but I grew up catching and eating hatchery early run individuals, and I appreciated the really dramatic difference between early and late run populations. And so I thought, well, if I'm going to target trying to understand the genetic basis of a phenotypic variation in salmon, I want to try to understand run timing. I want to understand, the gene or the genes that are making early run individuals early run. Okay, so this was about 5 years ago when I started collecting the samples for the study that I'm about to present to you. Okay, so this is the background. Now we're going to start getting into the study that I performed. All this data had been performed, but none of these studies have actually understood the genetic basis or evolutionary history of the genes that are actually causing early run timing, right? And so the goal was for me to identify the genes that make early run individuals early run, and then actually understand the evolutionary process that produced early run timing by analyzing the genes that were actually mutated to create early run timing. Okay? Does that make sense? Any other questions? Okay. So basically the study design for this is pretty simple. So what we did was we started with steelhead, which is one species that has early run timing, and we basically collected early and late run samples from up and down the West Coast. So from northern Oregon to central California. And basically these are really hard to get. There's very few early run individuals. And basically these are really the only populations that have significant early run populations. We try to get there's a couple places in Washington, but politics make it difficult sometimes to get samples that we want for our study. So for steelhead, we were only able to get early and late run populations from these locations. So basically we had 3 locations that have both early and late run populations, one of them being the North Umpqua, and then we had 2 populations. One of them only has early run, and one of them only has late run. Okay? And so basically what did we do? Well, we took these, and what's interesting about this is these are 5 locations from 4 distinct ESUs. So this is one ESU, this is an ESU, this is an ESU, and this is an ESU. Okay? So 5 locations from 4 distinct ESUs. And basically we got a hold of these samples and we just generated very large amounts of genetic information then. So we sequenced their DNA across the genome at a high resolution, and we analyzed that DNA sequence. Okay? So we started the analysis by taking all of the data, okay, all of the data from across the genome, and basically just performing a principal component analysis on, okay? And the 4 groups correspond actually to the 4 current ESUs, okay? So there's 4 groups here, 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 and here. Within any group, within each group, there's both run timings present. So when we have different run timings from the same location, they're in the same group. And in one group, there's 2 locations that are in one genetic group, okay? So basically, this is actually exactly so this is what people have seen for over 40 years, that different runtimes within the same geographic location are genetically very similar to each other, but different geographic locations are distinct. And actually, it supports the current, these 2 populations, they're actually very genetically similar to each other, and they're in the same ESU. It makes a lot of sense, right? They're not complete. It turns out we can actually separate them. So if we rotate this around and look at another axis, we find genetic differences between, in this case, this is the Siletz and this is the North Umpqua. But the Siletz and the North Umpqua, these 2 locations within the same ESU, are genetically very similar to each other relative to other populations from different ESUs. Okay? So our conclusion from this was that actually, even when we use large numbers of SNPs, basically data from across the whole genome, we still find the same pattern that people have been finding for over 40 years, where the overall structure relates to geography and not to run timing, right? So basically, genome-wide data supports the current ESU delineations, okay? Can't remember exactly what I have next, so let me see. Okay. So basically, though, I told you that the goal here was to actually identify the genes that are controlling runtiming. And so we said, well, what if we now take a population or a particular location where both runtimings are present, and we perform what's called association mapping to try to find regions of the genome that are associated with runtiming. So now we have hundreds of thousands of polymorphisms throughout the genome, and let's scan through all of those and look if any of those are strongly associated with runtiming, okay? So when people think about phenotypic variation in natural populations, they usually tend to think things are highly polygenic, okay? So meaning many genes are controlling a phenotypic difference that you see in a natural population. So when we went into this knowing that we're gonna try to find genes, but we might find multiple regions of the genome, and they each have a small effect. Okay, that's sort of what we were thinking when we went into this, right? But it turns out when we actually performed this association mapping, we were shocked to find that we found one extremely strong peak of association. So one region of the genome that was extremely strongly associated with runtiming, and nothing else in the genome was significantly associated. Okay. So in this particular case, so this is performing association mapping on runtiming in North Umpqua steelhead. We had hundreds of thousands of SNPs, and basically each dot here represents a SNP. And basically what's being done here is just a chi-squared test to test if the different SNP alleles are associated with different runtimings. Okay? And so basically for all the different SNPs, what we found is that so the higher you are up here, the more significant the association between the particular SNP and runtiming is. So basically what you can see is that we found lots of SNPs that were strongly associated, but they were all on this within this 2 megabase region of the genome, and nowhere else in the genome did we find a significant association out of hundreds of thousands of SNPs across the rest of the genome. Okay? So we were shocked that it seemed like there was a single gene, a relatively simple genetic basis that was controlling this difference between early and late run individuals. Okay? Now, we did this in the North Umpqua, and we thought, well, let's do this test in a different location. So the sort of current belief is that run timing is very evolutionary plastic, and it's evolved independently in different locations. So we thought, well, let's look at a different location and let's see, is it the same gene? Maybe a different gene is used in a different location, what's going on, right? And so we performed association mapping. This time we performed it on the Eel River. And it turns out we actually found a virtually identical pattern of association when we did this on the Eel River, Okay, so basically the same genetic locus controls early, so what do we know now? The same genetic locus controls early run timing in Eel River steelhead, as well as the North Umpqua steelhead. Okay, now, let me see what I have next, so I can, okay. So basically what's interesting, we said, well, what is doing this? You know, it seems like there's this single gene, this is the early run gene. Well, what is this gene? It turns out that the peak of association is right on top of and slightly upstream of this single gene called GREB1L, okay? And we'll talk about the function of GREB1L in a minute, but let me just say this. So we found the same gene was associated in both early run populations, from these two different locations. But the question is, well, there could have easily been independent genetic evolutionary events in the same gene that produce early run timing. It just turns out these mutations, these random mutations, happened in the same gene, and early run timing evolved using the same genetic mechanisms in different locations. Alternatively, maybe it's possible that early run timing evolved in one place, and somehow that new early run allele then spread around to different locations somehow, okay? So basically what we decided to do is we decided to reconstruct the evolutionary history of this locus in particular. So look at the evolutionary history of this locus across all the different early run populations that we had access to, okay? So basically what we did in this case, so just so you sort of know, to do all this association mapping and to do the PCAs that I showed you, characterized population structure, we used a method called RAD sequencing. But then to sort of focus specifically on this region, we actually amplified what are called these amplicons, specific sequences in this region, and sequenced them in lots of individuals. Okay, so we generated more sequence from specifically at this locus in all our individuals to be able to reconstruct the evolutionary history of this locus. Okay? So basically, we generated more sequence data at this locus specifically, and we built a phylogenetic tree. And what we found was really striking. We found that actually there's two monophyletic groups. So all of the late-run individuals, irrespective of where they came from, form one monophyletic group. And all the early-run individuals form another monophyletic group. There's some differentiation within the early run timing. But basically, this tree topology is completely inconsistent with the idea that there was independent evolutionary events in different locations. Okay? So basically what happened is there's a single evolutionary event that somehow happened that produced early run alleles, and then those spread around to different locations. Okay? So we'll continue to talk about that. How did that happen? But let me say really quickly, one of the interesting things that we found was we had a couple individuals that were originally, they were classified as, they were given to us as late run samples. But it looked like they were actually heterozygous, so they were at an intermediate position in this tree, and it looked like they were actually hybrids. They were half early run, they had one early run allele and one late run allele. And it turned out when we started looking more into the phenotypes of these individuals were caught. They were the earliest individuals caught within sort of the late run group, okay? So it looks like there's a single locus that completely explains early versus late run timing. And it looks like the late run allele is semi-dominant. Okay, so if you're heterozygous, you're more late run like, but you have somewhat of an intermediate phenotype between early and late run individuals. Now, we're interested in characterizing this more thoroughly. We only have about 4 early runs or hybrid samples that we found so far. But overall we were shocked at the simplicity of the genetic basis of such a complex behavioral and physiological adaptation. Yeah.
So I guess just I don't know much about fish spawning. They all spawn though still in the same area.
Yeah. So this is a great question.
Expect more hybrids to, I guess, occur.
Yeah. So let me show you. So basically, if you look at this, early run spawn. Here's, I don't know how well you can see this, but this is a bar of early run spawning and late run spawning. So basically, they're somewhat spatially and temporally isolated. So early run individuals tend to spawn earlier, and they spawn higher up in the watershed. Okay. But on some years, there is mixing depending on the condition. So for example, if it's relatively dry, it might be easier on, in some locations, and it depends on locations, these are complex things, but in some areas, under some winters, late run individuals can get higher up in the system and spawn with early run individuals, or in some years maybe the flows are too low and early run individuals can't get up high enough to be separated from late-run individuals,
So in
for
those kind
example.
of situations, it's possible for like a late-run individual to produce a hybrid, or like a early-run individual to produce late-run.
Yeah, well, if you have, so we'll talk about more that actually is important. The level of dominance
Yeah.
of this is important for sort of the long-term persistence of this allele, and we'll talk about that at the end.
Okay.
How do you know that the early ones were the later ones to later alleles evolved? Like, how do you know that they didn't come first?
Yeah, how do we know they didn't come first? That's a great question. What we know, there are ways to sort of infer that, but what we know is that if you look in a phylogenetic context, everything is late run, but early run has popped up in a couple species. Okay, so it's clear just putting in a phylogenetic context that early run timing is the derived phenotype. Now, we're doing something now, we're completely resequencing this region to actually identify the cause of the polymorphism. So we find an association, but the association doesn't mean the SNPs that we identify are actually the SNPs that are causing the phenotype. There's what's called linkage disequilibrium, where within a region you find statistically significant correlations between SNPs. So we've found SNPs that are correlated with the mutations that are actually causing early run timing, but I'm not claiming here to have found the exact mutations yet. That's what we're looking for right now. But we know we can reconstruct the evolutionary history of this region independent of knowing the exact causes of polymorphisms. Okay? So basically, so if we skip forward a little bit, or wherever, go back to where we were. Basically, so we have this, our data really strongly, how long do I have, Gordon? 11? Till 11?
Yeah.
Okay. So I wanted to make sure. I was giving a lecture the other day, guest lecturing, and I was like, we were talking about probability and genetics, and it was awesome. I was talking, and then I looked up, and I was totally out of time. And so I'm trying to be, it shocked me. I didn't realize at that time, so I'm trying to pay attention right now. Anyway, okay, so basically we have this, our data is completely inconsistent with the idea of independent genetic evolutionary events producing early run timing in different locations. But our data doesn't explain how you could have early run timing evolve in one location and then end up spreading around. Now, if you have good genetics intuition, it makes a lot of sense, given that they hybridize and all these things. But what's sort of the mechanism by which that happened? So basically, what I love about population genetics is that it's like having a time machine. Okay, so if you understand population genetics theory, everything that happens in a population influences patterns of genetic variation. Okay, so different demographic events, selection, all these different things that can happen in a population influence patterns of genetic variation in the genome and in specific regions. If you understand population genetics theory, you can actually look at patterns of genetic variation in a contemporary population and reconstruct what had to happen, the processes that happened in the population to produce the patterns of genetic variation that you currently have. So literally, even though you can't go back in time and see what happened historically influenced the current contemporary patterns of genetic variation. And so when you understand population genetics theory, you can reconstruct the past from contemporary patterns of variation. Okay? So basically what we did was we looked at a more detailed way at the patterns of genetic variation in this region to understand the evolutionary process of early run timing. Okay? And specifically, what we looked at were two summary statistics, and these are called Tajima's theta and Watterson's theta. Did you guys talk about those at all in here?
No.
No? Okay. So basically there's something called coalescent theory that's a very powerful logical framework for interpreting DNA sequence, okay? And these are basically two summary statistics that describe patterns of genetic variation in a region of the genome, okay? So let me just sort of explain this to you. So what we can do is we can look across the whole genome, and we can produce these two summary statistics. Tajima's theta, also called pi, average pairwise nucleotide differences, is basically an estimate of theta. It's this logic that this guy, Tajima, came up with in the early 80s. But there's another way is you can estimate this parameter, population parameter called theta, just based on the number of sites that are actually polymorphic. Okay, so you can look at the average number of polymorphisms between all copies of all chromosome copies in a population, but you can also just look at how many sites do you find polymorphic, irrespective of the frequencies, the allele frequencies of the different sites. Okay? But basically, if we look across the whole genome and we look at late-run individuals and early-run individuals, we find that these two summary statistics are very similar in late-run populations to each other, and early-run populations and late-run populations are virtually identical for both these summary statistics when we use the whole genome data. Okay? Now, when we look specifically and we summarize the patterns of genetic variation specifically at this GRUB1L region, so the region of the genome that controls early run timing, what we find is that when we look at late run individuals, Tajima's and Watterson's theta are basically identical. They're the same as each other, and they're basically identical to the genome-wide patterns. However, when we look at early run individuals, what we find is that we find dramatic reductions in both these summary statistics, and we also find that they're very different from each other. Okay? This difference is actually what's summarized in a statistic called Tajima's D. Did you guys talk about Tajima's D at all? Mm-hmm. It's a test for bottlenecks as well as a test for, basically it's a test for changes in population size as well as a test for selection. Okay? But basically what this is, what this tells us is there's what's called a very skewed site frequency spectrum. So the allele frequency distribution in this region, they're very different than what would be expected under neutrality. So this tells us, one second, Gordon. This tells us actually that the early run allele was under incredibly strong, this is a very, very strong signature of positive selection. Okay? So the early run allele, after it evolved, was under very, very strong selection. It was a novel evolutionary event that was very adaptively advantageous and selected for very strongly. And that positive selection is what facilitated the spread of the early run allele around to different locations. Okay? So if understanding population genetics theory allows us to see patterns and evidence of strong selection at a locus, for example. Did you have a question, comment, Gordon?
Oh, I was just going to mention something that might help students understand. We did talk about allele frequency distribution. We talked about bottlenecks causing a loss of rare alleles much faster than another index like heterozygosity, but this is analogous. But it's at a single locus, which is cool. You know, genome-wide and look at population molecules. Or here, you look at a single locus and see selection.
Yeah, so if you remember, I said that Tajima's D can be a test for bottlenecks or demographic events, but it can also be a test for selection. Basically, if we saw this pattern across the whole genome, what that would suggest is actually a very strong recent expansion of population size. But basically, we find most of the genome looks relatively neutral, slightly bottlenecked. This is slightly lower, and it's probably actually significant. I don't want to get into using coalescent simulations to, try to put, error bars around these statistics. But basically, the fact that we find it restricted to this specific region means that it's selection that's causing it, not population, not demographic effects that would influence the allele frequency distribution across the entire genome. Okay? So basically, the story in Steelhead is that there was a single evolutionary event where the early run allele evolved in some location. This is cool, so I want to mention this. Turns out, if you look at two things. This is in the Umpqua River, which is relatively far north, these summary statistics. And if you look back here, what you find is that this is the Siletz and Umpqua group within the early run. What we find actually is that there's a weaker signature of positive selection in more southern populations. So what happens is if you have what's called a selective sweep, a new allele arises and it goes to high frequency, you see that, but over time, new mutations accumulate on top of that new allele, and the signature of selection disappears over time. Basically, we have two pieces of evidence. One piece is that within the early run allele, the haplotype in Siletz and Umpqua is more diverged from late runtime, the late runtime haplotype, and the signature of selection in early and late run populations, in early run populations in this northern group is stronger. That actually suggests that the early run allele actually evolved in the south and has more recently spread to the north. Okay? So basically what we think happened is somewhere, maybe in Northern California, you could discuss this all day long, but basically somewhere, probably in the south, the early run allele evolved, and eventually that new genetic variation was very advantageous. It rose in frequency in those locations, but it spread, and it ended up spreading north because it was so advantageous. Just a small amount of that allele sort of leaking into other places ended up, that allele was under strong selection, and it ended up spreading and creating new early run populations, throughout the up and down the West Coast. But what happens is over time you have latent early run populations within a particular watershed exchanging genetic material, and most of the genome ends up becoming homogenized between early and late run populations within a given geographic location, except this allele, this particular locus that controls run timing. Okay? So basically, so GREB1L is a single locus that really controls this dramatic adaptation that's both physiological and behavioral in nature. And so what is GREB1L? Well, it turns out not that much is known about this gene, but it's been actually found in a couple association studies in humans. It's been found to be associated with obesity in Hispanic youth. And it's also been associated with polycystic ovarian syndrome in Han Chinese. It's basically a transcription factor that physically interacts with the estrogen receptor, and it's expressed in, let me say really quickly, the two main adaptations within early run salmonids are fat storage, so they store more fat to be able to survive for an extended period of time in freshwater, as well as delayed sexual maturation, so they don't have mature gametes when they enter freshwater. So their sexual maturation is on a sort of a different schedule with respect to salt water versus freshwater. So it makes sense that they would actually, this gene that's been implicated in similar, similar physiologies in humans could control such a dramatic behavioral and physiological adaptation within salmonids. And basically, it turns out this gene in mice is expressed in the hypothalamus. And it's known that the hypothalamus contains these key neuronal populations that drive feeding behavior and energy expenditure, basically metabolism and behavior. So it makes a lot of sense that this single locus could control such a dramatic difference. Okay, so basically the steelhead results were pretty clear, and so we thought, well, this adaptation of early run timing, like I told you in the beginning, you have early run individuals in both steelhead, but also in Chinook salmon. So in Chinook salmon, you have a very, very similar situation where early run individuals enter freshwater with immature gametes, they store a lot of fat, they migrate up, they oversummer in these deep pool pools before spawning at a somewhat temporally and spatially isolated from late run populations. Okay, so we thought, well, let's do the same experiment in Chinook salmon and see what we find, see what's the genetic and evolutionary basis of run timing in Chinook salmon. Okay, so what we did was we just did the exact same experiment. We actually got more locations, early and late run populations from, early and late run populations we're about to submit this for publication, so I'm like getting to the nitty gritty little details, and I thought one of these, my student had messed up one of those triangles, but no, it was good. Basically, we did the same experiment, but in Chinook salmon, we were able to actually get more locations. The problem is that many of the Chinook samples we get are from carcasses. So steelhead don't die immediately after spawning. They can actually return and spawn multiple times. Chinook salmon die, so a lot of the samples we get from Chinook are carcass samples. They're degraded. So Chinook, we got more locations and somewhat more samples overall, but it turns out a lot of the samples were bad. So it's not really any better than steelhead. But basically, we did the same thing. We started off by collecting genome-wide data using a method called RAD sequencing. we performed a principal component analysis, and we saw the same thing. We saw these different groups that correspond to the current ESUs. And within particular groups, you have both run timings. So again, the overall genetic structure relates to geography, not to run timing. Okay? But then we did the same thing where we said, let's perform association mapping to see if we can find a locus that controls run timing. And when we did that, we were shocked to find virtually the exact same thing we did in Steelhead. We found a very strong peak of association at GRUB1L, nothing significant anywhere else in the genome. We then looked at patterns of genetic variation across the landscape, and we found the exact same thing. So the same SNPs shifted in allele frequency in all the same populations, suggesting the exact same mechanism where you had a single evolutionary event at the GRUB1L locus that spread around to different locations. Now, there's two hypotheses here. It's something that would have been awesome if we would have found it. One possibility is that Chinook salmon and steelhead diverged 15 million years ago, okay, somewhere between 10 and 15 million years ago. One potential thing was that before Chinook and steelhead diverged, this early run allele had evolved, and it had been maintained in both lineages over time. But what we actually found is that the SNPs that we identified to be associated in Chinook and steelhead were different. And so actually, there's been two genetic evolutionary events. So once in Chinook salmon, the early run allele evolved and spread around, and once in steelhead, the early run allele evolved and spread around to different locations. So these were independent evolutionary events across 30,000 years, 30 million years, 20 to 30 million years of evolutionary time since these diverged. Okay? So basically, I think this story is really interesting from a basic evolutionary genetics perspective, but I think it has really profound conservation implications. Okay? And I want to sort of frame this a little bit in the context of Robin's quote here. So basically, previous genetic studies were correct with respect to phenotypic evolution. So you can argue that actually this phenotype did evolve in multiple locations. The allele only evolved once, and then that allele spread around and the phenotype evolved in different locations. But they were the previous interpretation was completely wrong with respect to genetic evolution. So actually there was only a single genetic evolutionary event, right? There wasn't parallel genetic evolution where, it's very easy to evolve this early run timing from a genetic perspective, right? And so basically, you can take Robin's quote, and if you modify it a little bit, you can say, the evidence for repeated parallel phenotypic evolution of run timing in Chinook salmon indicates that such a process is likely, provided that habitats capable of supporting the alternative life history trajectories are present and sufficient. Robust source populations, so robust source populations that contain the necessary pre-existing genetic variation are maintained. So basically, this mechanism of evolution is really great from a salmon perspective because any single early run population is dispensable, right? If you lose an early run population, well, you maintain early run alleles in other locations. So those early run alleles can stray, can end up leaking sort of back in and reestablishing an early run population in a place where that population had been extirpated, right? However, the problem, though, is that now we're basically at a point where this early run allele has been almost entirely extirpated from the entire range. So basically, we're at a point where we're about to lose this anciently evolved allele that has really important, cultural, economic, ecological benefits, and it's not protected. It's not protected at all. Okay, and the current framework does not, basically, the current framework of using overall genetic structure to define ESUs provides no protection for this type of basically this type of situation, right? So what are some of the, let's see what I have left. Okay, yeah, so the problem is that most source populations have been extirpated and none are robust. So basically, what I like about this story is that identifying the run timing locus led to completely opposite conclusions about the evolutionary basis and conservation priority of run timing variation. Okay, so basically, this No, this is right, sorry. I'm confused. Basically, run timing is completely genetically determined. There was a single ancient evolutionary event in each species. This new allele, this new early run allele was very advantageous and spread through positive selection and straying. The fact that we found bullshit to evolve early run timing suggests that the genetic mechanisms, so there's very limited genetic mechanisms by which this early run phenotype can evolve. It's not like there's thousands of different genes in the genome that could be mutated to produce the early run phenotype, okay? This tells us that if we lose this anciently evolved allele, it's very unlikely to just re-evolve. It's such a small sequence space that mutations have to happen in to create new early run alleles. I'd say it's early run populations that allow early run alleles to persist are an extremely high conservation priority, and this really demonstrates how using genomics and actually identifying specific alleles that are, specific loci and alleles that are controlling differences, adaptive differences that we're interested in preserving, really changes the way we think about these things. But it makes it actually very difficult to start thinking about it from a legal perspective. How do you begin to take this into consideration to decide, do you protect this allele? Do you call this allele a very significant unit? Well, that's probably not appropriate. At the same time, I don't think it's appropriate to continue what we're doing right now, which is providing no protection for early run individuals. And so I don't know what's going to happen with respect to how the policy will change and start to incorporate these types of data to actually make legal decision, to define units of legal protection with this new data that is genomics that's available that really allows us to, not just sort of guess about the evolutionary mechanism, but really determine in a satisfying way the evolutionary mechanisms that produced adaptations that we're interested in preserving. Okay, so let's see. Do I have any more slides? Oh yeah. So this was led by my student Dan Prince. He's started his third year as a PhD student, but another grad student, Omar Ali, and Hannah both contributed to the study. This is on the Sacramento River, just north of Davis, salmon fishing. What
species is that?
This is Chinook. This is a late run Chinook.
Wow.
It's 36 pounds. This is Sean Rourke. He's a research scientist in my lab, a permanent postdoctoral level research scientist that did all the molecular work for this and basically is involved in everything we do in the lab. And there was a number of collaborators on this study, including a lot of people worked hard to get these samples from various locations, people at different agencies and conservation organizations and universities and all this stuff. So thanks for listening to that. All right, so any questions? So how do you approach this problem? So I talked to Fred about this morning. He said, well, you have to figure out a solution to this. And I'm like, I'm not smart enough to figure out a solution to this. I can point out the problem, but coming up with a solution is much more difficult than pointing out the problem, okay? So I think this paper is, as far as I can tell, the clearest demonstration of the problem with the current methods of using genetics to delineate conservation units. But I don't propose an easy solution to dealing with this. We don't in the paper. Fred thinks we should, but he also, at the same time, he's saying the paper already has too much information and should be split into two papers. So I got feedback from Fred a little bit on this. But on the other hand, he hadn't even read the paper. He was just giving me feedback without reading it, so I don't really trust what he was saying. What do you guys think? Do you think do you understand this example and sort of how these new really powerful approaches have great potential to change the way that we delineate conservation units, but at the same time have potential to open up a can of worms because now we start thinking about, conserving specific alleles, and it could become unpractical very, very quickly. So it's very challenging.
Why did they think it was a parallel event at first then?
They think it was a parallel event at first because, view. Basically, because when you look at the overall genetic structure, what you find is that pairs of early and late run populations within the same geographic location are genetically very similar to each other. So sort of the idea here is that if this was your ancestral state and it moved to different locations and spread around, you sort of have to evoke multiple evolutionary events to produce a tree topology like this. But the problem is that populations actually don't have phylogenetic trees. Only single loci have phylogenetic trees, right? Different loci are behaving independently, and there's genetic recombination and all these things. So I think, from my perspective, that this was an overinterpretation of the data in the first place. But at the time these were made, we didn't really have the ability to characterize a lot of loci, and we didn't appreciate how distinct different loci throughout the genome can behave independently from each other with respect to patterns of genetic variation. Okay, so that was, that's sort of my justification for their overinterpretations. Yeah.
So you said at one point that you thought that this adaptive mutation, genotypes, this adaptive mutation had transported to other geographic locations,
Yep.
and then at that point there was enough homogenization among the populations, the extant population and the new mutant
Yep.
population, that's where the rest of the genome, that's why the rest of the genome shows none of those characteristics. But I'm thinking about super invasive alleles, like salamanders in California, where they're tracking those alleles in real time, and it's not. It seems like in that case, the allele is exporting faster than the rest of the genome, not that it's all going and then becoming homogeneous.
Yeah, so basically there's two potential mechanisms of spread that we can't test. I tend to agree with you on that. It's possible that this allele sort of spread in a heterozygous state, and the allele spread faster than the population. I think that's probably what actually happened. Another sort of a simpler way to think about it, if you're not a geneticist and thinking about different alleles moving, is that you have two summer run steelhead from one location that reestablish a new population in a different location, or not reestablish, but happen to return back to, summer steelhead are abundant, salmon at some frequency stray, they don't return to the river where they were born, and a couple of summer steelhead end up going to a new place. And they're really successful there, and they start a whole new population on their own. But over time, they mix with winter runs, and the winter run population probably has a bunch of alleles that are locally adapted, so they're adapted to this new specific river, and basically the rest of the genome from this new summer population ends up looking like the local winter population, either actively or passively because of numbers. So that's one possibility, but I tend to think that you're probably right. Yeah, no problem. No problem. But yeah, I tend to think that you're right, that the allele sort of spreads in a heterozygous state. But for people, it's more complicated to think about when people are sort of starting out. But we haven't tested between those two hypotheses. Yeah, we don't know the extent to which. I'll say a couple things on that. One thing is that if the early run allele was spreading really freely and easily, I would have expected these two different early run groups to be more homogenized. So I tend to think that there is pretty strong selection against the heterozygous state, and it's not that the early run allele is just spreading freely all over in the heterozygous state. And the combination of that and the fact that we have the heterozygotes seem to be the earliest caught late run individuals. From a conservation perspective, you actually want the early run allele to be completely recessive. If the early run allele would be completely recessive, it would even with strong selection against phenotypically early run individuals, the early run allele could persist for a long time. This Hardy-Weinberg equilibrium. You guys, I'm sure, have talked about this. But basically, the early run allele could persist for a long time in late run populations. But the fact that there is not complete dominance of the late run allele, it doesn't appear so, and the fact that it looks like the early run allele is not spreading, I'm not optimistic that the early run allele can persist for a really long time in the absence of early run populations. One of the things we're interested in doing is genotyping large numbers of late run individuals in different spatial areas to try to find what the frequency of the early run allele in different late run populations, and see if, as soon as we move away from any early run population, so most late run populations aren't close to any early run populations. My prediction is that we won't find any early run alleles in those. These are the types of things that we're trying to do now to follow up on some of this. So resequencing the whole region, we're actually interested, once we characterize the mutations, working with a colleague that studies obesity in humans and behavior and metabolism, and actually introducing these mutations into a mouse model and trying to understand the cellular and molecular mechanisms that this gene could actually use to produce such a dramatic physiological and behavioral differences in salmon, by using a mouse model to do that. I think this gene actually probably has has potential as a therapeutic target for metabolic disease, obesity, diabetes in humans, but there's a lot that we don't understand about it from a cellular and physiological basis of how it actually functions. So that's another route that we're going in with this. We don't do that kind of stuff in my lab, but with a collaborator at Davis. Yes, student first.
Do you know of any differences in fitness between early and late run?
Yeah, so basically, in most places now, early run individuals can't survive throughout the summer, so they basically die. In a few places where early run populations persist, I tend to think of it as that they're able to access habitat that's not really accessible to late run populations, and being able to access that habitat, their juveniles can do well and survive, and basically they can persist. And it's a little bit different. This isn't well understood, but my feeling is in steelhead, it's actually more about accessing habitat, because early run populations tend to persist in places where there's a really steep area that's difficult for late run individuals to ascend in high flows. But in Chinook, I think it has more to do with spawning time. So early run Chinook spawn in late September in a lot of places. In late September, water in the lower river is still really low, and fall run individuals cannot migrate into the river. It's lethal conditions in the lower river. So by migrating up in the spring Chinook salmon are actually able to, and so if they spawn earlier, their juveniles hatch earlier, start eating, they get bigger faster, they're more competitive. But the difficulty is surviving the summer to be able to spawn earlier. So my feeling is that in Chinook, it has more to do with the earlier you can spawn. As soon as rivers go up on the west coast, as soon as the first rains come in the fall, Chinook salmon just migrate up like crazy into these rivers and start to spawn. Like they're just waiting for there to be enough water and the conditions to be sufficient for them to be able to survive. So basically, I think it's advantageous for them to be able to spawn early, but you can't spawn early unless you migrate up in the spring, because the conditions are basically lethal due to flows at the end of summer being so low and it being warm, that kind of thing. So one of the other cool things about this is now we also, in addition to sort of understand this, we now have a really powerful tool to understand the ecology and to manage early and late run populations. So now we can do things like collect a whole bunch of juveniles from different spatial locations within a watershed, and we can figure out what habitat are the early run populations using, because we can genotype a juvenile now, and we can know if it's an early run or a late run, where in the past there was no ability to do that. So we now we actually just got a sample collection permit approved to collect like crazy all over 8 rivers in Northern California. So basically I'm a fisherman, that's why I work on this. And so we have like hook and line permits to sample in all these amazing places. And so we're going to do that to ask this question, to understand what habitat early run individuals are utilizing, and what are the advantages, ecological advantages of being early run. For example, are early run juveniles, do they tend to be larger? Do they tend to be exclusively in some habitats that winter runs can't access, and those kind of things? But basically, in the winter, there's a lot of water. It's hard to see and count fish, so it's hard to know where they go. So it's very difficult to do these types of things, and people don't really have a good feeling for how the different runtimes utilize the habitat differently, but now we have a really powerful tool to address that, because we can just phenotype any individual and determine its run origin. So yeah, that's something we're doing too. Yes, Gordon.
So have you thought about cryopreservation of sperm, saving that allele, and the possible downsides of that for the...
Yeah, someone said to me, they said, well, if the genetic basis of this is so simple, who cares about preserving the allele? Because we can just CRISPR it in the future. So it's so simple, we can just reconstruct this early run allele. And first of all, we don't know anything about the mutational events and how many mutational steps there are to create the early run allele, and so that's not feasible yet. We're trying to figure that out. But on the other hand, there is that argument that can be made. It is relatively easy to conserve an allele through things like freezing it or through recreating it in the future. How does that fit into the legal difficulties here? I don't know. But certainly, I would say, let's definitely cryopreserve these alleles if it gets to that point. But certainly we don't. I mean, it really actually is at that point already, but there isn't active steps being taken to do that. Basically, because there's not legal protection of early run populations, for example, in the Salmon River in the Klamath, it's the one location that still has early run population. Old growth is still, there's hardly any old growth in California, but old growth is still being logged. The tribes are still harvesting half of the wild early run individuals each year. And I don't blame them. They're not the ones who caused the early runs to be in so much trouble, but nothing is being done. Even though early run populations are independent management unit, they're treated as an independent population. Because that population is not legally protected, no real actions are being taken to prevent the extirpation of early run populations. No serious actions are being taken. Yeah.
So, Mike, the two lineages are pretty adaptively differentiated in a whole bunch of characteristics. Also things like thermal tolerance and disease resistance, a bunch of different traits. So
I don't think we actually know that very well. We don't know that. I expect that, but I don't think we. So you're talking about summer run versus winter run steelhead.
Yes.
You're saying they're adaptively differentiated at lots of different traits. We don't. I tend to agree with you, but we don't. That actually hasn't been. Peter Moyle has argued to me that I always say early run individuals are adapted to survive this extreme oversummering conditions. He says, well, they're just adapted to get to places where there happens to be a cold water refugia at a particular time. So we don't really know the extent of the adaptive differences other than the behavioral difference of different migration timing,
Sure.
as well as, so the behavioral differences of different run timing as well as this difference in gamete maturation. But I tend to agree with you that there are a lot of things. And when I say it's a single locus, well, it turns out we don't have, so addressing Ryan's question, there are probably other loci that are adaptively different, but they're much, the signal is much, much weaker. And if they're highly polygenic, if other phenotypic differences that are adaptive differences are highly polygenic, we don't expect to be able to even detect those differences. So you could argue, Ryan's argument fits in nicely to counter that argument that even if we conserve just this allele, that might not be sufficient to conserve early run populations in the long term, because there are other adaptive differences that aren't, that just conserving this allele or re-CRISPRing this allele wouldn't protect. But by far, I tend to think that polygenic, it's also, when things are highly polygenic, it's actually hard to purge alleles that are, that are influencing highly polygenic traits. So I tend to think at the same time, trying to decide if I want to bring up an animal breeding example. I won't. But I tend to think that those will persist for a long time, independent of early run populations persisting. But how long, I don't know.
Well, and you also think that there's a chance that this region's just massively pleiotropic?
I don't think it's. I don't think it's massively pleiotropic, but I do think it's a transcription factor in an estrogen pathway expressed in the brain, and it probably controls a lot beyond just the simple things that I know. But I tend to think like there are other adaptive differences at other loci that are polygenic that we're not detecting because our experimental design isn't designed to detect those types of things. But yeah, it's complex. I don't think that's the problem. So we rely on genetics because we can look at fish, and we can just see them, but it's really hard to know, like physiological adaptations. How can we quantify phenotypic differentiation in physiology? It's difficult, and it's not feasible to really understand how distinct natural populations are often by just using phenotypes. So we use genetics as a proxy for evolutionary significance and sort of ecological and evolutionary exchangeability, but clearly using basically connectivity and amount of gene flow between populations, we miss really important adaptive differences like in this situation. So basically using genome-wide patterns, or what I call in this paper overall genetic differentiation, is insufficient to recognize this screaming signal that exists. But now genomics allows us to better understand the extent of adaptive differences between populations and perhaps rely more on genomics to figure out what's actually adaptively distinct, irrespective of the connectivity between populations, which is basically what's reflected in the overall population structure. Yeah?
So you were saying where you come from, the early run were supplemented by fisheries, right?
By hatcheries?
Or hatcheries, sorry. So in a way, it is still being made, that allele is still being made in hatcheries. That's obviously not ideal.
Yeah, I don't think it's not legally sufficient, but it's also, for example, the North Umpqua River has, this is crazy, climate change. The North Umpqua River has both a summer steelhead hatchery and a spring Chinook hatchery, so the two early run populations. They've been doing a decent job. They kick out a couple thousand hatchery fish each year, or whatever. Adults come back. There's problems of rapid domestication selection and rapid domestication that reduce the fitness in the wild in salmonids. That's well known. But it turns out this summer, we sort of had this extreme drought, extremely warm. Turns out the water actually got too warm. All the Chinook salmon, all the spring Chinook salmon died at the hatchery. And for summer steelhead, or maybe it was the other way around, one of their pipes got clogged, and there wasn't enough flow, and all the fish died because of that. So certainly, in 1 year, they had an event where the entire year of both early run timing individuals died at that hatchery, like this last summer. So I don't trust the ability of hatcheries to be able to maintain that long term.
Fair enough.
Yeah, fair enough.
And basically, what I am optimistic about is that new habitats. So basically, as you go further north, the summer season becomes more and more short. So they can be moving throughout the whole year. So there's no advantage to be early run. So you lose, as you move into British Columbia, you lose early run populations like this. However, I tend to think actually a lot of new habitats will open up that will be potential early run habitats in the future. So even though maybe early run populations with climate change have no future in California, the longer we can maintain this allele in the lower 48, the more potential this allele has to be able to establish itself further north. And if I was the Canadians, I'd be pissed that we're not doing a better job protecting our salmon, because basically I had another paper where we show basically this is the mechanism of evolution and anadromous salmonids. Alleles evolve in one location and then spread around because populations are so connected. It's really amazing. What other species throughout the whole entire range of Pacific salmon throughout the Pacific Rim, from Japan to California, you have this really diverse area. But all these fish, and they're incredibly locally adapted because they have this natal homing, they return to their stream. But basically, they return, throughout the Pacific Rim, they're born in these little tiny tributaries. But they go to the ocean, they sort of overlap in the ocean, then they go back. So salmon are really interesting because you have this dynamic of extreme local adaptation with potential for gene flow across the entire species range in one generation. I think that's a really amazing and unique thing about salmon. But basically, because of that, the mechanism of evolution in Pacific salmon, based on our work, is this, where alleles evolve in one place and then end up being spread around. So basically, we really are compromising. So by letting populations become extirpated in one location, you're compromising the future adaptability of other locations pretty seriously. Yeah.
So do you know if you or others are intending to look at how far north these alleles have moved so far?
Yeah, it's hard to get samples. The thing that limits us is getting samples. Basically, there's a really famous steelhead fishing river called the Dean River in British Columbia that I'm trying to get samples from that potentially has a early run population. As you move further north, it's pretty interesting. Actually, there's sort of something analogous. As you move sort of further into southeast Alaska, in some locations you have early run populations, but what's interesting is they migrate up in the fall and they overwinter and then spawn in the spring, and late run populations come up right after sort of the snowmelt and spawn right away. So we're interested in getting those. I tend to think, it might be the same gene, but I tend to think, although it's sort of superficially similar, it's different enough that the alleles, maybe it's GREB1L, maybe it's not, maybe historically it was the same evolutionary event, but I tend to think those alleles wouldn't be exchangeable, but I don't know. That's something we're investigating.
Is the allele frequency high enough that you could just broad scale test for heterozygotes?
That's what we're interested in doing that, but again, we're doing that in California. The bane of my existence is getting samples. I can genotype thousands and thousands, so we just developed a new method we call Rapture. We can genotype 10,000 samples trivially. If I can get 10,000 samples, I can genotype them, no problem. But I can't get 10,000 samples to genotype. Okay, so I would like to do that. I am always trying to get samples, but getting the samples is challenging.
But you don't necessarily need an early season phenotypic.
No, I know.
run.
Yeah, I
so
can just
go
yeah,
to the
no,
grocery store and buy a bunch of
but
British
yeah, it's not
Columbia
just for
salmon.
biological reasons, but it's political reasons that it's difficult to get samples.
Yeah, I gotcha.
Yeah. Yeah.
So for the political reasons, it's just a bunch of people that don't know science, but they try to talk science to you.
It's a lot. Yeah, there's that, but there's also just other geneticists who work on salmonids in other areas that think that getting us their from my perspective, there's an infinite number of questions to ask. There's way more questions to ask. I mean, we just talked about, like, I could spend my whole career following up this. Okay? All of the questions that come out of this, I could spend my whole career on, right? Many, 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 many questions. There's basically way more questions to ask than are possible to ask. So I get really annoyed when people are reluctant to provide samples because basically it's not a competition at all. There's way more questions to ask than there is time or people to answer, than to actually to do. So it frustrates me that people basically for competitive reasons don't want to provide samples because they're salmon geneticists and they want to do it, even though they think about these things from a very different perspective. We're the only one thinking about this question like this and approaching it with these methods and this. But as soon as they hear, they say, oh, well, we're studying summer and winter steelhead in other contexts. We don't give you our samples, things like that. And then there's like international issues where we're trying to get these Canadian samples, and we fortunately have some Canadian collaborators that we're working with to try to get these Dean River samples. But they had obtained some Dean River samples before at some point. But it turns out when they obtained the Dean River samples before, they were just sport fishing, and they caught the samples, and they told someone, but they didn't. It was sort of a miscommunication, and so now they're like the regional managers who are not research biologists are mad at these guys for like getting samples in the past, and it's just all this shit that just makes it hard to get samples. It sucks. because it limits our ability. Now with new methodology, the money to do these experiments, the time to do these experiments, none of that limits us. It's just our ability to acquire the samples. So that's what frustrates me the most about working in this field.
Did you ever think of like going off the grid and just collecting these samples?
Yeah, we do some of that. But
I'm just fishing.
yeah, that's what we do. I mean, basically, but even if we fish, even if we sport fish, and we can even do this. If I can catch a fish legally, I can handle it.